Welcome everyone to Science Society, and of course a special welcome to you, Takeo, for taking the time to come here.
Thank you.
And before we start, let me give the audience a short introduction, and then we'll do like a conversation to maybe get to know you as a scientist a little bit better. So, Professor Takeo Watanabe, he did his PhD in psychology at the University of Tokyo, and he taught at Boston University before then starting his position at Brown in 2012, where he is now a Fred M. Seed professor, and he is in the Department of Cognitive, Linguistic, and Psychological Sciences at Brown University. And in his research, he focuses mostly on perceptual learning and the roles of attention and reinforcement signals in learning. And yeah, it's such an honor having you here. And I think it's such an important research field. So, how did you discover kind of a passion or an interest into like going into research? Was it something, a childhood dream, or did something later on happen that sparked your interest?
I don't know.
That's fine.
So, yeah, I usually do whatever I like to do. So, by my intuition. So that's all.
Yeah, that's good though. I mean, to follow your interest, it's a good thing, right? That's wonderful. So I'm glad you had the opportunity to do that.
Thank
Um,
you.
so, and then how did you become interested in going into this specific field of learning? Um,
Yeah. So as you said, I'm specialized in perceptual learning. However, my interest is learning and memory in general. However, the perceptual learning occurs upon a visual stimulus, which is very clear to everybody. So, you, so there's not much difference among people what is perceived, as as opposed to, for example, memory, which has a huge standard deviation across subjects or people. So that's why I thought that to understand learning principles, um, it would be a kind of the fastest way to learn perceptual learning mechanisms.
yeah, I also think that that's a really interesting field and topic, and we had a few guest speakers, but that were not so precisely, in this field, but related to. So it will be really interesting to discuss after you're
Yes.
presenting your work and the slides, I think. So yeah, thank you for giving us kind of your story about what motivates you to do this research. And yeah, so for everyone, the slides are pinned on top of the room, and yeah, the stage is yours. Thank you.
All right. So am I supposed to start now?
Yeah, if you would like. Yeah,
Okay. All right. Okay, good. So first of all, thank you so much for inviting me to give a talk in such a great opportunity. Um, so I have to apologize that I actually I catch a cold and sometimes I need to cough, so that could be kind of noisy. And also, I also need to apologize that this cold, made me develop a very strange Japanese accent in English, which I usually speak perfectly. I'm just joking.
yeah,
Okay.
it's the same with me. It was COVID made my accent.
Okay. So Today, I'd like to talk about efficient learning in children with rapid GABA boosting after training. All right, please go to the next slide. So, if you look at websites or read books, the statement that children learn better than adults or children has more capable of learning things more efficiently than adults. These statements are overwhelming, right? How so? I have been interested in perceptual learning basically in adults. Then, when I saw these statements, and I checked scientific evidence for that, there's hardly any. That may be a little too strong, but there is not many papers focusing on this question. And there's hardly any neuroscientific research to systematically investigate whether and how children learn more efficiently than adults. That's why I decided to address this question, okay, using the method I have used. Okay, so actually, we come up to the hypothesis that one of the reasons why children learn more efficiently than adults is because children stabilize learning rapidly. So what is stabilize or stabilization? Please go to next slide. You see the kind of weird pictures, right? So, so to understand the concept of stabilization, let me talk about a kind of a specific paradigm, okay, for retrograde interference. So at the top, you see 3 yellow dots, okay? The central dot is on the right to the top and the bottom dots, right? So your task, so in some trials, this stimulus is presented. In other trials, the stimulus in which all the dots are aligned straight, okay, are presented. And your task is to indicate whether the dot is actually aligned to the other dots or on the right, okay? To exaggerate the stimulus, this stimulus is made to be, I mean that the distance between the 2 dots and the central dot is very much exaggerated. However, the true distance is very, very small. So at the beginning, hardly anyone can tell whether the central dot is on the right or aligned straight, okay? But as a result of practice of maybe hundreds of trials, subjects can learn to discriminate between the 2 situations, okay? That is a perceptual learning of S1, okay? However, if you look at the bottom, the middle one, as you see, there are 2 stimuli, S1, S2, okay? Assume that training of S1 occurs, all right? Then, immediately after the end of training of S1, you are asked to practice on the stimulus S2. Only difference between S1 and S2 is whether the central dot is on the right side or on the left, right? So if these 2 trainings are conducted without any time interval between the trainings, what happens is that learning of S1 disappeared. Only learning of S2 is observed. So this indicates that learning of S2, okay, disrupted learning of S1. That is called retrograde interference. All right? However, if you look at the bottom figure, if there's a time interval of a few hours between the two trainings, then learning of S1 and learning of S2 are observed. In that case, no retrograde interference is observed. So this experimental result indicate that right after learning of S1, okay, the status of learning S1 is plastic or changeable or fragile. Okay, so while this learning status is very plastic, if training of another stimulus occurred, then the previous learning is destroyed, okay, and overwritten by learning of the new task. Okay. However, if a few hours passed after the training, the learning gets stabilized. Okay. Then after stabilization is complete, even if another learning occurs, the old learning stays. Okay, that is the concept of stabilization. So if you go to the next one, entitled plasticity and stability changes. All right. So if you look at, you see the plastic and stabilized. Okay. Please ignore red ones. Okay. So as a result of training, okay, the degree of plasticity is enhanced, right? And then after that, the plasticity gradually gets down, okay, to the status of stabilization. So this a few hours process is called stabilization. Okay, if some other learning occurs during this stabilization process, then the original learning is destroyed or disrupted. Okay? So, coming back to the children's issue, we assume that we hypothesize that children may more rapidly stabilize learning. Okay? That may be a reason why children learn more efficiently than adults. Okay? So GABA actually plays an important role in stabilization. This is a well-known fact. Okay? However, there's a problem because it is also widely known that children have an immature GABA system up to 24, 25, or 26 years in age. Okay? So this indicates that the concentration of GABA in children's brain may be lower than adults' brain. So this means that these two statements appear to be contradictory to each other, right? So children may rapidly stabilize learning. To do that, GABA is necessary. However, the amount of GABA in children is maybe smaller than that of adults. So how can we reconcile this? So to address this question, we conducted an experiment to measure the amount of GABA. I mean, more precisely speaking, the changes in the amount of GABA, okay, before, during, after training with adults and children. Go to the next slide, please. And then next slide, please. So I talked about visual learning. That is my specialty. Okay, visual process. So visual. Do you understand visual? Yeah. So that's a challenging word to a Japanese. So in my class in Brown University, I usually say that when I say visual, this means visual in any case. So visual perceptual learning refers to long-term improvement of a visual task as a result of visual experience. Okay. So there are some mechanistic differences between the training of visual perceptual learning and some other types of learning and memory. However, okay, in post-training processing, this indicates that the processing after training, okay, mechanisms of this post-training processing is generally similar between visual perceptual learning and many other types of learning and memory. Okay, maybe one of the most famous post-training processing is consolidation of learning during sleep. Okay, although I do not go to this consolidation during sleep. In any case, so this indicates that if we study post-training processing using visual perceptual learning, we may get a result which could be generalized to some other types of learning and memory. Okay, please go to the next one, showing magnetic resonance spectroscopy. So for this purpose, okay, we used magnetic resonance spectroscopy, by means of which usually glutamate as an excitatory neurotransmitter and GABA as the chief inhibitory neurotransmitter are measured. Okay, but here for the purpose of this experiment, we actually focused on the measurements of GABA because, as I said, GABA is known to play a significant role in stabilization. Okay, please go to the next one. So in the first experiment, we employed 13 children and 14 adults. So maybe some people interested in memory or high-level cognition would think that the numbers of subjects, children, number of adults, could be a little too small, but it's not. That is an advantage of doing research of perceptual learning. Usually, perceptual learning and motor learning gives much smaller standard deviation in results across subjects than in many other types of learning and memory. Okay, that is one of the reasons I like visual perceptual learning. Okay, if we go to the next slide, showing the brain, the region, enclosed by the yellow circle, okay, not circle, yeah, okay, yellow square. That is a region belonging to what is called early visual area. Okay, early. Do you understand my pronunciation? Early versus late. Okay. A friend of mine, American friend of mine, told me that I should name my son Earl so that I will practice early in everyday level. In any case, my son's name is Louis, not Earl. So please go to the next slide. So this is the procedure of the experiment. Okay. So before the training, GABA measurements were made, and also after the training. Okay? It's a very simple paradigm. Next slide, please. So this is the training task. Okay? This is a very famous training task in vision science for detection. This is called the detection task for orientation detection task. In each trial, the two stimuli are successfully presented. Okay? One of them contains orientation. In this particular case, the first stimulus contains orientation. The second stimulus contains only noise, right? And in half of the trials, a stimulus containing orientation is presented at first, followed by noises. In the other trials, the stimulus containing orientation is presented in the second place. The subject task was to indicate which interval contains orientation. Okay? Since I made the orientation appear to be conspicuous here, but in the real stimulus, there are much more noises, so that it's very difficult to tell whether there's any orientation structure in the stimulus or not. But by doing practice, you will be able to tell or detect orientation easily. Okay? So this is learning to detect orientation. Okay? So that is a task, and this task was repeated, task performance was repeated around 800 or 900 trials in the usual case. If you go to the next slide, which shows the results of behavior improvement by training. Okay? As you see, improvement occurred for both children and adults. Also, if you go to the next slide, the amounts of improvement for the two groups are comparable. There is no significant difference, right? Then if you go to the next slide, which shows, okay, the amount of GABA in early visual areas for the pre-test and post-test, okay, before and after training. In adults, there's hardly any change in the amount of GABA in adults, right? If you go to the next slide, this shows children's result. So as you see, there's a huge boosting in the amount of GABA after as compared with before the training. Okay? So of course these boosting are significantly larger in children as compared with in adults. Then please go to the next slide. Okay, so if you go to the right, okay, so I think the F is this is kind of left over from the graph in the paper. Okay, so no other meaning. Anyway, sorry about that. If you go to the right, and if you see pre-training, okay, the blue bar represent the concentration of GABA in children, the red bar for adults. As you see, the concentration of GABA before training was indeed much smaller in children than in adults. That is consistent with a widely spread knowledge that children's GABA system in the brain is immature, right? However, if you look at the children's concentration of GABA, pre-training, training, and post-training, As you see, it increased gradually, right? And rapidly. Gradually and rapidly sounds a little contradictory. I've said that, so gradually would be the better word. Sorry. And then if you go to the left graph, okay, this indicates, okay, more detailed time course of GABA changes in children and adults after training. So 0 in the x-axis represents the offset of training, okay? So as you see, in adults, hardly any change in the amount of GABA. However, in children, there is a rapid change, okay, in the amount of GABA, right? So in this experiment, we found that children increased the GABA concentration in early visual areas during training. However, no such increase was observed in adults. So this suggests what happens, okay? So in children, please go to the next slide, which indicates a schematic figure, schematic graphs for GABA changes in children and adults, okay? So A, B, C are different trainings, okay? So when training A occurs, the concentration of GABA rapidly increases in children. And then when training B occurs, the same thing occurs. When training C occurs, the same thing happens, right? On the other hand, if you look at the adult graph, okay, when training A occurs, perhaps GABA level could change a little bit, could increase a little bit, but not noticeable. But with a larger scale like ours, the GABA level may increase like this. Actually, that's what we found in a previous study with adults. Okay, so it increases, but it takes 2 or 3 hours to get an increased concentration of GABA in adults. So if you look at that, if you compare these two, it's obvious that more training of different tasks can be done in children than in adults. That is why children may learn things more efficiently than adults, in the sense that children can learn more items within the same amount of time than adults. All right? Because unless stabilization occurs, okay, if during stabilization after learning A, if training B occurs, then learning A could be destroyed, right? So you have to wait for a few hours to learn something new after learning of an original item, okay, in the case of adults. But in children, okay, no such waiting is necessary, okay? However, we just measured, okay, GABA concentration. Is this really beneficial? I mean that the rapid change in GABA really beneficial to children's learning or not? To check that, we need to do a kind of behavioral experiment. So next slide, please. You see the slide before, right? So this is a concept of stabilization, okay? It's a paradigm to indicate how stabilization occurs. So I talked about retrograde interference, right? So this means that if stabilization is complete, there's no retrograde interference. So this indicates that the amount of retrograde interference is inversely correlated with the amount of stabilization, right? So the retrograde interference is a good index for stabilization. I mean that inverse index. So please go to the next one. So as I said, retrograde interference is inversely correlated with stabilization. And in our experimental results, the rapid increase in the GABA concentration is likely to lead to rapid stabilization. If that is the case, children may become resilient to retrograde interference with a shorter interval between two trainings than adults, meaning that children can learn things, okay, within a shorter interval between two trainings, okay, without getting any retrograde interference. Okay, that is our hypothesis. So we conducted the experiment using 14 children and 14 adults. So go to the next one. Next one is the procedure. Okay, you see the two trainings. Okay, schematic procedure slide. So although in the first experiment only training A was conducted, but here there are two trainings being conducted. Training A and training B. Okay, training A represented in the red rectangle, and training B represented by the blue rectangle. Okay? And time interval between the two trainings was said to be 1 hour. Okay, 1 hour is, so as I said, stabilization starts immediately after the offset of training, and it takes a few hours to get stabilization complete. So 1 hour is a kind of in the middle way of stabilization. This indicates that there should be some retrograde interference occurring. Okay? Please go to the next slide. So task was the same task as in the first experiment. Okay, task A, both task A and task B. The difference between task A and task B was the difference in orientation trained, with the procedures otherwise exactly the same between the two tasks. Okay, in task A, this orientation was presented, and the orientation task A, above task A, and in task B, a different orientation was presented. Okay, there are actually two orientations are perpendicular to each other. Okay, so please go to the next slide, which shows the results. Okay, so this is the percent of improvement. Okay, so if retrograde interference occurred from training B to training A or learning A should not occur, whereas learning B should be observed. So that is a case in adults, right? So although this result is not really strong, it may be because the time interval between the two tasks was 1 hour. That was stabilization in the middle, right? In any case, this, statistically speaking, this shows consistent result with retrograde interference in adults. Please go to the next. So that is retrograde interference. Okay, please go to the next one, which shows children's as well as adults' result. Okay, in children, okay, not only learning B, but also learning A were observed, indicating that there was no retrograde interference in children. Okay, and this is a significant difference between adults and children. And then please go to the next slide, which indicates experiment 2B. Okay, so then we conducted the experiment in which the time interval between the two trainings was as short as 10 minutes. Okay, so actually the purpose of this experiment was to use the result as a control result, meaning that children, maybe 60 minutes there was some difference, but in 10 minutes children actually showed, both children and adults would show retrograde interference. Then to find out how rapid or how long stabilization takes for children. Okay. So next slide, please. Next slide shows the training procedure. Only difference between the previous and the current experiment is that the time interval between the two trainings was 10 minutes, okay, instead of 60 minutes. So this is the next slide shows the result in adults. Yeah, as predicted, since time interval was such a short time as 10 minutes between the two trainings, very clearer retrograde interference pattern was observed in adults. Namely, there's no improvement of task A, whereas there was significant improvement in task B. That is a very, very clear pattern of retrograde interference, meaning that training of B interferes with learning of A. Okay? And that is an indication of stabilization incomplete. Right? If A was stabilized, then there should be no retrograde interference, right? So this next slide, please. So this indicates retrograde interference. Very clear pattern of retrograde interference. Please go to the next one, which shows children's result. To our surprise, with such a short interval as 10 minutes, children was free from retrograde interference. That's surprising. This indicates that, I would say that this suggests that, please go to the next slide, the statistical difference is huge. So this result suggests that, okay, children actually got stabilized within 10 minutes, all right? So we know that it takes at least 3 hours for adults to get the learning stabilized. There's a huge difference between children and adults, right? So this is the conclusion of getting all the results together. First, the first experiment indicates that concentration of GABA in early visual areas in children is smaller, indeed smaller than in adults. That is consistent with a widely spread notion that children's GABA system in the brain is immature. That's why the concentration of GABA in children is indeed smaller than in adults. However, during training, the concentration of GABA rapidly increases in children, right? In contrast, the concentration of GABA in adults stays fairly constant, at least during time we measured GABA changes, right? And then we conducted an experiment to check whether retrograde interference occurs in children and adults. And we found that retrograde interference occurred in adults with 10 and 60 minutes of the time interval between the two trainings, whereas in children, no retrograde interference occurred in these intervals, even with as short as 10 minutes of time intervals between the two trainings. So this suggests that, as I said, maybe stabilization in children occurs within 10 minutes, okay? Whereas stabilization takes around 3 hours or more in adults, according to our previous experimental result, which I did not show. So basically, these results suggest that a rapid increase in the GABA concentration in early visual areas of children makes children stabilize learning so rapidly that children can learn more within a certain time period than adults. Okay, so children can learn more items without learning being interfered with each other, right? However, in the case of adults, they have to learn one item, and then they need to take time to learn another item. Okay, that is not necessary for children to do. All right, that is supported by the rapid GABA increase after training in children. Okay, that's it. Thank you very much. Oh, okay. I need to tell the acknowledgement. Okay, so these people are involved in the study. Okay, the Sebastian Frank, during pandemic time, Sebastian Frank. Okay, we are not allowed to do any experiment in Brown University. So Sebastian went back to Regensburg in Germany. That's his hometown. And he asked his ex-advisor, the professor of University of Regensburg, Mark Grundy, he's American actually, to let him use the magnetic resonance spectroscopy tool. And then he talked to Ulrike Frank, who was the headmaster of an elementary school of University of Regensburg. And she was very much persuaded about the importance of the research, and partially because she was Sebastian's mother. Anyway, so we got a subject and experiment in Regensburg. We had a plan to do this experiment in Brown, but in any case, and then the Yuka Sasaki is another professor in Brown University, and Marcus Becker is an undergraduate student in University of Regensburg. Andrea Q was an undergraduate student of Brown University. They are all involved very effectively. Thank you very much.
Thank you so much for presenting your study here so well. It's so interesting, especially to me, because my PhD thesis was very general, because I did a lot of like different collaboration, but it was the role of the inhibitory system on memory and learning.
Mm-hmm. I see.
And back then, the inhibitory system was so understudied, so I was very excited to see this, that highlights the importance of inhibition in the circuitry, how important it is for learning. So it's really interesting to me.
Thank
But
you.
yeah, but also in general, as you mentioned in the beginning, that there are so many claims made, especially from very famous people, right, that write books, and then when you discuss people say that you're crazy because you say there's no evidence. Did you have that experience before? It is just interest I
Right.
have. There's this research of flow, for example, right now for concentration, all these things that people write books about and say they're researchers, but there's no real hard
Actually,
evidence so
right.
much.
I would say that they have evidence, under the observation of children in school and in that level of evidence. However, there was no clear neuroscientific evidence for that.
Yeah, exactly, and how it exactly works.
Hmm.
So yeah, I agree.
Hmm.
So, yeah, this is really interesting. So, probably you get asked this a lot. What can adults do to keep the GABAergic system up? Like, would it be possible for people to take some sort of um, maybe drug that inhibits the uptake of GABA after learning, and then adults would be able to also learn more
Yeah,
frequently?
yeah, that could be one way. However, if you take GABA, as a drug, the GABA would be effective all over the brain, right? So in that case, I'm not quite sure how much effective um, the stabilization would be. So there should be some areas or some networks in which glutamate or some other excitatory neurotransmitter being effective for stabilization to be successful, although GABA plays a very important role in stabilization. So I think one way of doing that, although not yet developed about that technology, is to by by using online neurofeedback technique, a certain areas GABA concentration can be controlled. Um, so that could be one way to improve learning of adults. I mean that online neurofeedback is like a biofeedback of which was developed a long time ago. So if you measure the concentration of GABA at a certain area, say early visual area, like we did, and then that the amount of concentration is shown to a subject in a real-time basis. Then as a subject, if subject we try to make the concentration of GABA larger, if it happens to get larger, then, for example, circle showing the concentrate, representing the concentration of GABA in that area is enlarged or something like that. If it is and then amount of money to be given to a subject will be in proportion to the average size of the circle or something like that. So that is kind of similar to biofeedback in which a subject can control their body temperature in some way. So by doing that kind of neurofeedback method is advanced, it is not impossible for for us to control the concentration of GABA in a certain area of the brain. That could be one of the possible ways. As a matter of fact, I have conducted online neurofeedback using fMRI. However, the problem is that um, I, using both signal, which is measured by fMRI, um, neurofeedback is possible because you can take the both signal amount on on the real-time basis. However, the technology has yet to be developed for MRS signals to be shown on real time. So there's some time lag between the measurement result to be shown and the real real concentration changes in the brain. So this time lag needs to be much shorter in, with the technological advancement. If that came true, it is possible that we will be able to control the amount of GABA in, in a certain targeted area of the brain. That is important not only for younger adults, but also older people, because it is well known that the concentration of GABA in older brains uh, gets smaller than younger adults. That makes learning less efficient. For example, you cannot ignore noises with a small amount of GABA, right? For example, if you take your grandfather and grandmother to a restaurant, they really would like to go to a very quiet restaurant because they are not really good at ignoring noises. This may be because of the smaller concentration of GABA. So if more GABA is released, then they can concentrate on things that can enhance learning and or their intelligence level. So I think that the controlling the amount of GABA in the future is one of the most important things in the brain science.
Yeah, that's really interesting. And related to that, did you think of in the future to check for children with learning disabilities if
Mm-hmm.
their GABA
Mm-hmm.
response after learning is different too? Could be both ways, right? Some people maybe, some kids
Yeah.
learn a lot very fast, other, cannot, control also like an ADHD or autism
Yes.
spectrum disorders where they cannot, you know, cancel out noise and other sensory information.
Right. And some of mental issues came from imbalance between excitatory and inhibitory neurotransmitters, and some came from, for example, a significantly small amount of some neurotransmitters, right? So I think what neurotransmitters needs to be adjusted or controlled um, depends on the type of mental issues. Um, yeah, certainly I would say that, for example, in the case of ADHD, um, the larger amount of GABA would play an important role in improving some aspects of ADHD. I'm a kind of I'm suffering from a slight attention deficit, like many researchers. So that would be nice if that kind of technology will be developed.
Yeah, me too. I can focus very well on things that I'm very interested in, but if I'm not interested in it, like bureaucracy documents, I have a hard time reading them and focusing.
But
So,
I think that in a sense, attention deficit people tend to have more creativity than some other people. That may be because of the less cognitive control. That sometimes cognitive control is not best for creativity because you have to, behave or think, uh, with following rules, right? So that's maybe why there are so many great scientists who have some attention deficit, tendencies.
yeah, I agree. I theorize the same thing. There has to be a certain amount of chaos in the system to come up with some new ideas
Mhm.
that you don't control, and some compulsion disorder, so you keep trying, right? You have to be very stubborn and compulsive to learn certain things. required for scientific, work,
Actually,
I think. So,
right.
yeah, it's very interesting.
I find that we share something very important.
Yes. And I want to hand over the microphone to Dr. Shah. Welcome. Please go ahead.
Yeah, thank you so much for your wonderful talk. And
Oh,
my
thank you very
question
much.
from you, so based upon the last slide that you just demonstrated, we see a couple of the chemicals include the glutamate, creatinine, and
Yes,
choline, as well as the
right.
aspartate. So when we are looking for the spectrum, and that was very interesting to me in comparison with the GABA, and based upon the group that you just chose from the children 3 to 11 years old, I
Mm-hmm.
was just wondering to ask you about the correlation between the pattern of the chemicals as well as the kind of for those children struggle with the focal epilepsy and seizure, because when we are talking about the GABA in another side, we might should consider other factors. That was my first question, if you have any opinion over.
Actually, I have done basic research, so I do not know much about clinical cases. That is first, and also MRS measured only the concentration of glutamate and GABA, not other uh, transmitters. So I mean that there might I believe that, for example, about basic mechanism of learning, I believe that even stabilization, okay, is determined by not only GABA but also some other types of neurotransmitters. However, because of the limitation of MRS, it is difficult to indicate uh, whether and how much these other neurotransmitters play roles in stabilization. So similarly, it's get even more difficult to indicate that some other how and whether um, other neurotransmitters play a role in some mental diseases. That is based on my personal knowledge as well as technological limitation.
And is there any specific reason that you chose, for example, 3 years old, not infants, included in
Oh,
the research?
not infants. Actually, that's what we are trying to do. First of all, that's a great question. The reason why we did that was kind of the environmental restriction, and so and also so far no research group has conducted any research using MRS with infants, except for one group in Yale. Um, so we are trying to learn from them how to do that with infants. That's a great question. Um, it would be very interesting to know how stabilization occurs and how GABA concentration change occurs with infants. That's a great question.
Also, do you think that just adding some phenotypical, for example, measurement, such as, for example, you um, mentioned about the cognitive, or we can just talk about gait measurement and intelligence as well, can be beneficial if you want to add it into to your research or not?
Yes, I think so. But intelligence, I mean that there are so many aspects determining intelligence. Is that your question? Uh, so we just focused on learning, um, and also learning perceptual learning. So although we think that visual perceptual learning result can be extended to other types of learning and memory, which are more related to intelligence than visual perceptual learning. Um, we are not 100% sure whether the result can be 100% extended to other types of learning until, for example, the scientists in these fields will try to replicate our results. But I'm kind of optimistic about that because it is because post-training processing is quite common among many different types of learning and memory. However, about other product, so so there are many aspects determining intelligence. So the learning abilities is one of them, right? Also, the thinking things rapidly, more rapidly, is regarded as a result, is a manifestation of a higher degree of intelligence. And also the cognitive control strength is another part of another aspect of intelligence. So these different aspects of intelligence may be determined in different subserved by different mechanisms, and different mechanisms may be driven by the combination of different types of neurotransmitters and also the connectivities between different areas. So yeah, that's a great question. However, I've said that our research is only at the beginning of opening just open up some possibilities, but that's all I could say.
So good luck for your future research,
Thank you
and
very
thank you
much.
so much for your answer.
Thank you.
Yeah, thank you. Do you still have time for two more
Yes, of
people?
course.
Okay,
Yes.
great, thank you. Vicky, please go ahead. In case you're away from your phone maybe right now, I'll switch over to Kyle. Kyle, do you want to go ahead? How are you?
Wonderful. Amazing presentation. Thank you so much. I'm going to look into this a lot
I hope you
deeper.
understand my accent.
Yeah, yeah.
Okay.
Um, I was just curious, what sort of like practices or dietary measures are you um, currently implementing in order to boost your own GABA? I would imagine that you
Yes.
might have
Yes.
thought of this.
That's a great question. So yeah, that had been a big problem in the measurement of MRS because particularly, but not really GABA, but glutamate. The concentration of glutamate is greatly influenced by the time interval between. I mean the time passage after taking a meal. So we usually conduct the measurement strictly 1 or 1.5 hours after a meal, so that the 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 dietary the processing or nutrition in the body will not significantly influence the GABA level. But I was less worried about, as I said, less worried about the concentration change in GABA than the concentration change in glutamate in that case. Did I answer your question?
Absolutely.
Okay.
And very good point. And thank you
Okay.
so much. It was amazing presentation. Definitely might be sending you an email or two to ask a few questions in the
Of
future.
course. I'm happy to answer any question. Please do not hesitate to send me emails.
Thank you so very much.
Thank you.
Yeah, thank you. And I wanted to check with Vicky. Vicky, do you still have your question? Yeah, go
Hi.
ahead.
So I wasn't aware about GABA before I joined the room, and while you guys were having a conversation, I navigated through certain nuances. I'm a voice coach from India. I'm using a lot of neuroscience in my technique. I was reading that GABA is something that helps in relieving the stress. Whatever little knowledge I have about neuroscience, amygdala is also something that does the same with regard to understanding the anxiety and all. Is there some direct correlation between the amygdala and the GABA?
Amygdala, you mean you're talking about amygdala? Actually, I did not hear you very well. Kind of the voice is some vague, yeah, there's some vagueness. So I'm sorry about that. So did you ask about the amygdala and GABA?
Yeah.
Okay.
Is there some connection there?
Okay, amygdala is amygdala is a kind of area for anxiety, which could maybe whose malfunction could cause some depression or some post-traumatic memory. Um, I do not know about the relationship between GABA amount and the amygdala because amygdala is not my area of specialty. Um, as a neuroscientist, I should I, I should tell I should be able to tell you more about that because it's brain. However, I do not really know the role of GABA in the amygdala. Sorry about that.
Right.
I
And
can,
when
I
we are
can tell
given
a
the
little bit, but maybe it's outdated because as a PhD student, I worked in amygdala and GABA and memory. So
Oh,
actually, very interestingly, the amygdala suppresses a lot of activity to improve the signal to noise ratio, to kind of in the moment that it needs to record basically a field memory to be very efficient and very precise. And there, but in general, there have been recordings that people with anxiety disorders have a high activity in the amygdala, which makes us think that the GABAergic system is weaker in people with anxiety disorders. And yeah, there are different receptors transmitting and then changing norepinephrine effect. So norepinephrine is like a hormone that gets released during stress and acute anxiety that then makes the memory to be stronger and more ingrained in the system. So it's it's not very simple, though, all the interactions, and they are not 100% known, but in general, during regular state, people with anxiety have high activities or weaker inhibitory system. But then when a real anxiety or fear or threat signal comes in, um, the system gets suppressed very well to get like, so it's very interesting because it kind of correlates, Takeo, to your studies that GABA is very important to learn efficiently and to keep the memory, which it kind of correlates there. I think that's interesting, and a lot of those processes, I think, in those more old brain regions don't change so much maybe during age, but it would be interesting to do the same study and see differences in people that have PTSD and other disorders related to anxiety and measure the same way.
so I'm reading the title. It's mentioned efficient learning in children via GABA boost. A GABA boost is something where we are tweaking the natural flow of learning. So is it advisable?
oh, you're asking.
Yeah, I think we answered a little bit the question, right? Do you want to address it again, Takeo? Or?
Okay, actually, um, I I did not hear
Oh, yeah.
you very clearly. So if you repeat the question, that would be
Yeah,
helpful.
if a GABA boosting would improve basically memory.
Is it advisable because you're tweaking something in the brain? and you are coming in between the natural learning process of the children, right?
So
So is it
yeah,
advisable to use these kind of boosters for children education per se?
yeah. GABA boost. I would say that the GABA boosting is helpful to stabilize learning. However, okay, the GABA boosting should not occur during training. So during training, the brain needs to be flexible and plastic. In, in order to do that, glutamate, that is an excitatory neurotransmitter, plays an important role. So the combination of glutamate, that is an excitatory neurotransmitter, during training, and GABA boosting after training is necessary. Okay, that kind of two combinations are important for improving learning, or for creating the keeping improving learning. So that in that sense, GABA boosting is important in improving memory and learning. However, that is not all. I mean, glutamate boosting during training is important for learning acquisition. So in short, excitatory signals like by good enhancement in concentration of glutamate is necessary for acquisition of learning, and inhibitory processing which occurs with GABA is necessary for consolidating learning. Am I answering your question?
Yes,
Okay, great.
but I'm not aware which countries are using it nowadays. The country from where I come from, we don't use any kind of booster with regard to learning.
Uh,
For any kind of learning or training, because I'm a coach, I've been training people. We never use any boosters.
are you training people?
Yeah, I'm a voice coach.
Oh, I see. So, but you used some chemicals?
Nothing.
Okay, I see. I see. So
Only the
you,
hard work. The only chemical is hard work. That's it.
oh, I see. Okay, I see.
So, so are there some countries which are propagating this? Because we are not aware.
I see. So I actually I would say that it it depends on, that's a kind of difficult question, because I personally I do not recommend that any chemical uh, should be used to normal people. Normally, no, the people whose brain functions normally and whose learning ability is normal. I don't want, I don't like any chemicals to be used just to improve learning with normal people. If so, because there could be some side effects for that.
Hmm.
Yeah, that's my personal opinion. Okay, so if training is made to improve learning without relying on chemicals, that would be much better, in my opinion.
Yeah.
However,
My opinion
there,
too. Yeah.
yeah, there are cases in which just behavior training is not sufficient for some cases. In that case, the cases of some clinical diseases and clinical related malfunctions or dysfunctions, or uh, the or due to aging, um, if necessary, the, the chemical may play an important role, but only in
Mhm.
these cases. That's my opinion.
Thank you, doctor.
You're welcome. Thank you.
We have one more person, Einar, if you have time, and because I don't want to overstretch your patience. So I don't know if
It's
you could.
okay.
Okay, thank you so much. I appreciate it. Einar, go ahead.
Yeah, hi, Katerina. Hi, everyone. Um,
Hi.
just a quick question. I came late in this room, but uh, does glutamate um, is it like centralized, yeah, or does it work in actually
No,
the same way all over uh, the the brain here when it comes to neurotransmitters, or
yeah,
does decay? You know what I mean? Thank you.
yeah, because yeah, actually, I would say that the glutamate is effective almost all over the brain. Yeah, so is GABA. It, am I answering your question?
Um, yeah, kind of. But like if this GABA, this is like the glutamate is the neurotransmitters, right?
Yeah, that's
Neurotransmitter,
right.
right? And does it affect the synapses when you go out of the CST, like further out of the central system, or do you have any question?
Yes,
Any,
actually, usually synaptic efficacy uh, is associated with changes uh, in many neurotransmitters, including GABA. Uh, so, and also the GABA amount, GABA actually is is related to glial cells and also interneurons. So there are several different sources of GABA, GABA changes. And so we usually question when I talk about GABA amount changes is that is it related to glia cells or isn't it related to inhibitory interneurons development? So I say that this may not, at least the amount of GABA changes which are correlated with learning in performance is related to either synaptic efficacy changes or inhibitory interneurons, not glia, because glia is not supposed to do function related to learning. So to answer your question, the GABA is, I would say that the GABA is almost ubiquitous in the brain.
thank you.
Thank you.
I yeah, I don't study this subject, so yeah, but thank you very much.
You're welcome.
Well, thank you so much for coming here and presenting your research and answering so many questions
You're very
today.
welcome. I
This
really enjoyed, chatting with you and the audience.
Well, thank you. Well, that's very nice to hear because it you know it should be good for both, for us, but also for you. It should be fun. And um, I'm so glad that you do this research, that we finally, get to know what's the mechanism of learning and differences in aging. So yeah, I'm very curious to continue following your work in
Thank
the
you.
future. And we wish you all the luck and a lot of collaborations and funding for this. I think it's really important. So thank you so much for doing this work.
Thank you so much.
And yeah, enjoy the rest of your day, everyone. Also, thank you for coming, asking questions. And if you like discussions like this, we'll have another talk on Friday. Diffusion art or digital forgery. It's big data analysis and ethics around AI, the new AI platforms. It's a really interesting, was a really interesting publication and very hardly discussed topic. So yeah, if you want to join us on Friday, I'm looking forward to hear you all back. And thank you so much again. It was such a
Oh,
pleasure
thank
to
you very
talk with
much.
you. Okay, I'll close the room in three,
Okay,
two, one. Bye everyone. Thank
bye.
you.